are going to discuss an important question from CSIR NET June 2023 Shift 1. The question is, in mature erythrocytes, the end product of glycolysis that contains the carbons of glucose is option number 1 ethanol, 2 pyruvate, 3 acetaldehyde, and fourth one lactate. The correct answer is option number 4 lactate. Now we can discuss the answer in detail. What is glycolysis in mature erythrocytes? Mature erythrocytes, which is also known as red blood cells, show a unique type of metabolism compared to most cells in the human body. Their metabolic pathway is specially adapted for efficient oxygen transport. The most important feature of mature RBCs is that they lack mitochondria, nucleus, endoplasmic reticulum, ribosomes. Because mitochondria are absent, RBCs cannot perform aerobic respiration such as TCA cycle, oxidative phosphorylation, and electron transport chain. Therefore, they completely depend on glycolysis for ATP production. Let's look into the energy metabolism in RBCs. The main function of RBCs is oxygen transport through hemoglobin. To maximize oxygen delivery, RBCs avoid consuming oxygen themselves. If mitochondria were present, RBCs would use oxygen for oxidative phosphorylation as well. Oxygen delivery to tissues would decrease. Hence, absence of mitochondria is a physiological adaptation. Glycolysis in RBCs. Glycolysis is the pathway in which glucose is converted into pyruvate in the cytoplasm. The overall reaction is like this. Glucose gives 2 pyruvate, 2 ATP and 2 NADH, and the products formed are 2 molecules of pyruvate, net gain of 2 molecules of ATP as well 2 molecules of NADH. ATP produced through glycolysis is the only source of energy in mature RBCs. Here is the flowchart of glycolysis in RBCs. Here is the fate of pyruvate in most cells. In normal aerobic cells, pyruvate enters mitochondria and converted into acetyl-CoA by pyruvate dehydrogenase. And the acetyl-CoA enters the TCA cycle and large amount of ATP is generated through oxidative phosphorylation. But this pathway cannot occur in RBCs because mitochondria are absent. Now, let's look into the fate of pyruvate in RBCs. Since pyruvate cannot enter mitochondria, it undergoes reduction to form lactate. And the reaction is catalyzed by an enzyme called lactate dehydrogenase or LDH. And the reaction is like this. Pyruvate plus NADH plus H plus gives lactate plus NAD plus. And thus, pyruvate is converted into lactate and NADH is oxidized back to NAD+. Therefore, lactate becomes the final end product of glycolysis in mature RBCs. Regeneration of NAD+ is an important factor in this glycolysis because during glycolysis, NAD+ is reduced to NADH, and continuous glycolysis requires regeneration of NAD+. And the conversion of pyruvate to lactate oxidizes NADH back to NAD+, allowing glycolysis to proceed continuously. Without NAD+ regeneration, glycolysis stops, ATP production ceases, and RBC survival becomes impossible. Let's look into the ATP production. Although anaerobic glycolysis produces much less ATP than oxidative phosphorylation, it provides sufficient energy for RBC functions such as maintenance of Na plus or K plus ATPase pumps, preservation of membrane flexibility, maintenance of biconcave shape, as well as prevention of osmotic lysis. Why RBCs lack mitochondria? This is an important question. Absence of mitochondria is actually advantageous for erythrocytes because RBCs do not consume the oxygen they transport and maximum oxygen delivery to tissues is ensured. Thus, RBC metabolism is specialized for oxygen transport efficiency. Here is a table that would help you to revise the features of mature RBCs. Let's look into it. Mitochondria is absent in RBC, TCA cycle, and oxidative phosphorylation does not take place in this RBC. And the main source of ATP in RBC is anaerobic glycolysis, and the final glycolytic end product is lactate, and the key enzyme that acts in it is lactate dehydrogenase. Let's analyze the options of our question. First option was ethanol, which is an incorrect answer because it is produced during alcoholic fermentation in yeast, and human RBCs do not produce ethanol. And the second option was pyruvate, which is also an incorrect answer because it is the intermediate product of glycolysis. And in RBCs, it is further converted to lactate. And the third option was acetaldehyde, which is also an incorrect answer, and because it is also an intermediate in alcoholic fermentation and it's not produced in erythrocytes. And the last option was lactate, which is our correct answer, and it's because it is the final glycolytic end product in mature erythrocytes, and it also contains a carbon derived from glucose. And that's the end of our discussion of this question, and I'll be back with another CSIR net question. And do like, subscribe and comment.